All right, hello everybody. We're going to go ahead and get into today's lecture, which is going to be on photosynthesis. Just a couple things before we get going. First, this is one of the more complex topics in Unit 3 in that there's a lot of details that you need to remember, a lot of different protein names and things like that. I don't expect you to memorize all of it by the end of this lecture. I just want you to kind of get an understanding of the fundamentals. And so that way next week when we do models, it can make more and more sense and so you feel fully capable of answering any questions on it. Okay. You'll also notice that today's lecture looks a little different. There's more text and there's also videos that play along as well. I think it's important that we kind of watch what's going on as it goes. So that's why I've incorporated these throughout the PowerPoint. So that being said, let's go ahead and go back to the beginning and get started. So the sun has always been around since Earth's been around, and it's going to be around for another 5 billion years or so. And what's nice is that the sun can essentially be thought of as an endless source of energy, in that it's constantly giving off solar energy at all times of day, depending on what part of the Earth you're on. But we can take advantage of that solar energy and be able to convert it into a chemical form that organisms can use. Now, before photosynthesis, which is the process of converting solar into chemical, organisms used to be chemosynthetic, which essentially means they break down high energy molecules in their environment and then live off of that. But the first photosynthetic organisms, which could use solar energy, evolved about 3 billion years ago, and these were prokaryotic organisms. So one of the first species of prokaryotic organisms that could do photosynthesis were cyanobacteria. You can see an image of them to the right here. They evolved about 2.7 billion years ago. And essentially, they caused a huge disturbance in the atmosphere of the Earth. Because what they did is they took that CO2, which used to be very abundant in Earth's atmosphere, and converted it into the oxygen-rich environment that we have today. We refer to this as the Great Oxidation Event. Our atmosphere is approximately 21% oxygen, and most of this comes from this Great Oxidation Event that occurred. So the atmosphere we have today is thanks to this process and to these cyanobacteria. Now, prokaryotic photosynthesizers eventually became eukaryotic through endosymbiotic theory that we talked about in the last unit. If you need to kind of recover that, feel free to watch that video. It's one of the last videos in the last unit, or feel free to look at your notes. The main thing that you need to remember is that the prokaryotic photosynthesizer formed a symbiotic relationship with another cell, which is the modern day chloroplast that we see today. So here's that chloroplast right here. Again, some of the evidence for the endosymbiotic theory include the fact that it's double membraned. For today, the main organelle that we're going to be looking at within the chloroplast is going to be the thylakoid, because this is where the light-dependent reactions are going to occur. There are light-independent reactions, which means they don't use light, but we're going to talk about those another time. All right. So as I said before, there are going to be two main steps to photosynthesis. You've got light-dependent reactions, and it's exactly what it sounds like. These are the reactions that require light from the sun, just like we see there. And then there's the Calvin cycle, also known as the light-independent reactions. These are reactions that occur without light. Okay. Now I've got the chemical formulas here for the light-dependent reactions and the Calvin cycle. Please make sure you write these down, and by the end of this lecture, hopefully the light-dependent reactions will make much more sense. Okay, so let's go ahead and get into the first part here of the light-dependent reactions. So this is going to take place in the thylakoids. So if we go back to our chlorophyll here, in these thylakoids here, more specifically, it's going to take place in the membrane. You can kind of see the outline of it here, right? This is where this reaction is going to occur in the chloroplast. So let's go back to this one here. Now, the first major protein we're going to talk about that's in the thylakoid membrane is going to be photosystems. And there are two photosystems, photosystem 1 and photosystem 2. They are out of order in that photosystem 2 comes first and then photosystem 1 comes second, which is a little confusing. But the only reason that exists is because that is the order that they were discovered. Photosystem 1 was discovered before photosystem 2. And what these photosystems are going to do is they're essentially going to capture solar energy from the sun and then transport it from one protein to another throughout this membrane through something called the electron transport chain. And this is going to have a lot of effects that go with it, and it's going to set up the creation of glucose later on. So within the photosystem, there's going to be a molecule known as a chlorophyll, which you can see right here. And this chlorophyll is essentially going to serve as a house for electrons. Now, these electrons are going to come from water, and we'll look at that later. But, play that one more time. This chlorophyll that has the electron in it, what this chlorophyll allows to happen is when that solar energy comes in, which we call a photon, it's going to excite this electron and give it energy. So we can see photon of light comes in, and then the electron becomes excited because it's storing a lot of energy. It is then going to be passed on to the next protein, which is the cytochrome complex. So let's go back here. All right, so this is where we left off in our video. So you can see this electron, it's being passed on to the next protein here. Now, like I said in the previous slide, actually I could play this, this electron is going to come from water molecules. So you can see a water molecule here. It is split into oxygen and hydrogen, okay? Now, the hydrogen atom is where the electron is actually going to come from. So the hydrogen is split into a proton, which is left in the lumen, and the electron is passed to the chlorophyll to get the photon of light and essentially pass that energy on. So next, that excited electron is going to move on to the cytochrome complex, and it's going to create something called an electrochemical gradient, which you should remember from Unit 2. So here, we're going to use some of that energy from photosystem 2, and we're going to move hydrogen ions against their concentration gradient, which means we're using active transport. So what you can see here is we're essentially stacking hydrogen ions on this side, hydrogen ions just being protons. We're stacking these protons or hydrogen ions on this side in order to create an electrochemical gradient. And the reason we're doing this is because we're essentially storing potential energy that we can later use to make other types of molecules. All right, so once we have created this hydrogen concentration here, the electron is then going to be passed on through the electron transport chain to photosystem 1. And photosystem 1 is essentially going to do the same thing as photosystem 2. So it's going to sit here. It's going to wait for a photon of light to come in. Hopefully it doesn't take too long here. All right, so it's back in the chlorophyll, and boom. So you can see the electron now gets more energy from the photon thanks to the photosystem. Okay. Now this high energy electron from photosystem 2 is going to be passed on to these proteins or these enzymes that are free floating in the thylakoid stroma. So you can see it keep going. So here we have a molecule called NADP+. All right. Now we're going to add an electron to it. This is referred to as a reduction process, as you might remember from chemistry. When you gain an electron, it's a reduction process. So this NADP+ is going to gain an electron, and that's going to become NADPH, because it's also going to take a proton from the environment as well. So we have NADP+ plus an electron plus a proton. It's going to give us NADPH. So this is going to be a high energy molecule, and we're going to use this later to create glucose. But this is one of the main products of this light-dependent reaction. Ooh, go back up here. There we go. So we fast forward, and we got NADPH. Okay. Now, as you remember before, we were stacking hydrogen ions on this side to create an electrochemical gradient. And there was a purpose for that, because now we're going to take that potential energy that we've been storing up here and convert it into kinetic energy. Okay, so here's our hydrogen ion concentration. And what they're going to do is they're going to flow through a protein complex known as ATP synthase. So it's an enzyme because it ends in ase, and it's synthesizing ATP, so ATP synthase. And what this ATP synthase is going to do is as these hydrogen molecules come through ATP synthase, it's going to take this energy that's being converted from potential to kinetic, and it's going to create ATP molecules or energy molecules. So you can see all that stuff flowing through until the concentration is even, and ATP is going to be synthesized. So we can take ADP, add a phosphate group, ATP is created. Okay. Now we're done with our light-dependent reactions, and we have two products. We have NADPH and we have ATP. So those are going to be our two high energy molecules, which we're going to use later to create glucose in the Calvin cycle. Now, I'm not going to go over the Calvin cycle today. I think this is more than enough to learn for one day. So feel free to watch this again and again if you need to. I really like the ending of this video here too, because you can kind of see the whole thing kind of playing through. But make sure you write down questions if you have any, and then we will go over them on Monday next week.